So today is 9/25/17, September 25th. And we're talking about DNA. Does anybody know what that stands for? Somebody said nucleic acid. Oh, what is it? What is it? What's the word? What's the whole word? D something. Okay, so we've got the D and we've got the nucleic. Try this out for size. D. Somehow the recording got paused. So deoxyribonucleic acid, not important that you know how to spell that or remember it, but, you know, potential bonus question someday. What is DNA? It is, actually I should start with this. It's a long, stringy molecule. Something like this. Let's see. Here's a big mess. There, have some more mess. Yay, mess. Yay, string. Yay! Big long thing. Yeah, just keep it going. That's fine. I go to yard sales. I buy yarn. Oh, look at that. Yeah, look at that. It just keeps going. It's the gift that keeps... Oh, that's fantastic. You guys spontaneously decided to pass it around. I love it. You guys are awesome. So, yeah, just keep it going. See how far we can get this around the room. So we've got this long, stringy mess of a molecule. We could tie ourselves in complete knots with this. Huh, that's pretty cool. How do we get from this mess to something that actually makes you have a cowlick? Or me have a crooked pinky? Or somebody have abnormally thick mucus on the inside of their lungs? Or somebody have blood that doesn't clot correctly? How does that happen? Again, that's the big question we're going for. So let's go into a little bit, and I realize that this is, okay, we can just drop the DNA string wherever we are. I'll pick it up later. You don't have to draw this in detail. Here's what I want you to know about the big structural image of DNA. It is often referred to as a double spiral. And that's because, and that's a really bad double spiral. My first one was better. That's because there are two matching strands. You can imagine a ladder that got twisted. So a ladder to which someone took the two ends and went vroom, 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 and twisted it. That's what the DNA molecule looks like. Now, you notice that in here I drew a bunch of little colored stuff. DNA is actually pretty simple. So we're going to go to the more detailed picture of what it is. It's really made up of 5 things. 5 things. So we have what's often called the sugar backbone, and you don't need to know anything more about the chemistry than it is called a sugar. It's not like table sugar, sugar backbone, but we call the sort of outsides of the ladder the sugar backbone. So that's the first component of DNA. And then the rungs of the ladder are made up of 4 chemicals, only 4 chemicals. So 4 chemicals make you different than another human, and different arrangements of those 4 chemicals make a ladybug different than a maple tree. That's kind of amazing. It's like being able to write the entire works of William Shakespeare using 4 words and have it make sense. It's kind of astounding. So the 4 chemicals other than the sugar backbone are adenine, thymine, cytosine, and guanine. 4 chemicals. And we usually say ATCG. And here's the thing. They only ever interact with, they can only couple up in very specific ways. It's like a key in a lock. Who has ever stuck the wrong key in a lock? Okay. Or in, well, just yesterday I tried to start the truck with the car key. It didn't work. The ignition key wouldn't even go in, let alone turn. So they only fit in particular ways. It's like a lock and key arrangement. And adenine only pairs to thymine. Cytosine only pairs to guanine. And we're going to look at a good mnemonic for remembering that. Apples fall from trees, cars park in the garage. It works. So I'll let you go ahead and draw that. The letters can go in any order. They can end up in any order, but anytime you have an adenine on one side of the ladder, you're going to have a thymine on the other. Anytime you have a cytosine on one side of the ladder, you're going to have a guanine on the other. And you can have a long run of one letter. You can have a space where there are no two letters that are alike. But really, it's that simple. It's 5 components. It's a sugar backbone and these 4 what are called bases. And what you just learned is called the base pairing rule. It means that they only pair in specific ways. This is the base pairing rule. Interestingly enough, the base pairing rule was a really important piece in terms of people understanding, scientists who were working on the structure of DNA, in terms of them understanding the fact that there were these couplets, these pairs, because they found that, you know, however much adenine they had the same amount of thymine. However much guanine they had the same amount of cytosine. So logically, they inferred that they must be paired together somehow. Okay. That's about as simple as something could be, isn't it? It's not that complicated. And yet this allows all the living things we see, all the living things we don't see, you and all the bacteria that are covering you and all the bacteria that are on your desk and in your water and on your coffee cup. No, that's just me. In your guts, and a blue whale, and an earthworm, and a mouse, and a maple tree, and a dandelion. Those 5 basic chemical blocks, the sugar backbone and the 4 bases, allow for this amazing diversity of life when they're arranged in particular ways. Now, I don't know about anybody else. This sounds utterly bogus to me. This sounds not possible. When I really think about it at that level, like there's no possible way that 5 chemical pieces could allow for all this stuff. It defies belief. So the thing that we're going to keep digging for is how it works. I mean, I'm telling you it works, but why should you take my word for anything? Seriously. I could lie to you. I could make up a whole day's worth of absolutely bogus information and give it to you. Probably wouldn't do that, but you never know. I'm a little bit unpredictable sometimes. It does, I mean, those 5 basic chemical pieces interlock to form all the living stuff or to allow for the formation of all the living stuff we see. But how does it work? Oy. That's actually a question for tomorrow. We're going to start to talk a little bit about how it works. Now, remember, this stuff gets copied over and over and over again. And then somehow this stuff allows for the formation of living things, such as you and maple trees and blue whales and earthworms and ladybugs and bacteria and grass and dandelions. That's not a lot of notes. That's okay. What you're going to do now is if you have your textbook, you'll use that. If there's at least somebody at your island with a textbook, you can reliably share on this. So if you're already taking notes in Doceri, I'm going to ask you to make a separate project. So X what you're doing and create a new project. Does anybody need help with that? This is an experiment. So you're going to create a new document. I have often in the last 10 years had biology students replicate some of the drawings from their textbook. For people who are real visual learners as well as for some of the kinesthetic people, it's useful to copy those illustrations. This time, for the first time, I'm trying to have you do it on Doceri instead of on paper. We'll see how it works. You guys are my guinea pigs. So there are pictures on page, and it should have popped up on your classroom by now. Did it pop up on your classroom yet? The assignment? Okay. There are pictures on page 185 and 186. There are 3 image sets. There's figure, I'll take the DNA back. Thank you, sir. Yep. There's figure 10.1, figure 10.2, and figure 10.3. You're going to replicate those images. You're going to label them. So 10-1. I'm doing my best bad drawing here, as I recall it. You're going to color them, And you're going to write the caption, because the picture doesn't make a whole lot of sense without the caption. If you don't have a stylus, I have a few stylus pens I can loan out. You can trade back and forth. And do you know the trick about writing neater in Doceri? Zoom in, and then write your caption, and then you can shrink back out, because it's much easier to write neater when you're writing bigger. So if you zoom way in, you can write the words. Yes, ma'am. Yeah, you got first dibs. So you're going to be working on those illustrations. You will then export it as a PDF, and you'll submit it to Classroom. While you are doing that, Mr. Maple and I are going to start doing those one-on-one meetings with you, and you'll get to see your test grade if you took the test. If you still need to make it up, you need to make sure you get to that. Okay, so go ahead and get started working.